everyone. I'm a PhD student in Metrics Lab in King's College London, and today I'm going to introduce the cortical surface analysis to you with specific focus on the benefit for the functional study. I know that traditionally the neuroimaging studies are all conducted on the volumetric data using volumetric pipelines. So here I first plan to motivate you with the cortical surface analysis. I will introduce the free surface pipeline. I plan to talk about the surface registration, which is specific benefits for the functional alignment. And then I will give you some practical examples of the Workbench command and Workbench view demos. So you might be familiar with the volume representations of the brain, which gives you slices from the sagittal, coronal, and axial point of view. And surface actually starts from this volume data, where you first, from a structural modality that can clearly show you the tissue contrast around the cortex. It could be T1 for the adults and T2 for the neonates. So if we have a close look at this, the surface is actually needs to first fit in the inner surface and the outer surface of the brain cortex. Then you will find that it's actually a set of points interconnected to each other to form a triangular mesh. And on this mesh, just not like the voxels in the volumetric data, the surface also have its own unit. We call it vertices. That is the point on this mesh. And we can, each of the vertices can represent a number of cortical features that helps you to project any volumetric data, like the modalities can reflect the brain functions on it, like the resting state function networks or the task functions or the paths on these 2D cortical sheets. Then you can do the rest of the downstream analysis on the 2D cortical sheet here on this mesh. Why do we want to look at the data on the 2D mesh? Because the human brain cortex is actually a thin green matter layer. Because of this intrinsic 2D topology, it's very intuitive for you to visualize everything on the surface. You can see that in the early brain observational studies, the skin draws of the cortical areas are usually conducted on the surface or projected on a flat view. More than 100 years ago, Rodman first scanned the cell bodies in the histological sections across the entire cortex, and he managed to segregate the brain into a number of functional specific regions. And here, from then on, there are lots of studies trying to refine it and trying to improve these parcellations. You can see that these parcellations are presented on this kind of surface view. You can also see that these areas, these function-specific areas, there are in a 2D spatial arrangement, but not in a 3D field. For example, if I want to refer to the area 45, I would say it's on the inferior frontal lobe. It's between area 47, 44, and 46. I'm not going to just give you an XYZ coordinate in the volume space in this external coordinate. What is also interesting is that these cortical features themselves, they are not arranged randomly. So spatial adjacent body parts are represented into the adjacent field of your brain. You might have seen these funny cortical homunculus before, and you will see that the brain regions requires finer motor controls or more flexible controls, more sensitive areas like face or hands, controls your fingers. They are usually occupying more space on the cortex, and the less sensitive areas like trunks or legs, they are occupying less areas in the cortex. And these all shows that these functional areas, they are not arranged in a random way, in a coincidence, but actually they're trying to build connections and help you to efficiently process the signals of the sensory motor function. So all of these evidence showing that the functional regions, they are organized in a 2D cortical sheet, and it makes a lot of sense just to model the way it should be, but not to put them into an external coordinate. So there are already some evidence showing that just by putting the volumetric method to the surface can improve the spatial correspondence of cytoarchitectonic areas. Why would this significant improvement happen? The first problem of the traditional volume-based spatial normalization is that it's trying to push the constraint on the intensity, and it's trying to do the calculation of the distance in the Euclidean distance. You can see that it's not respecting the geometry of the brain cortex. So, for example, the brain activities that could be very close to each other in the spatial map, in the space of a volume, actually they can be far away from each other in terms of the surface. And now if there is only a slightly misalignment in the volume data, like this, in the orange and the blue, they're aligned to different sulfur bonds together, and then probably you are going to compare to completely different functional areas in the brain. And this will cause a lot of uncertainty and error in your downstream research. Now let's have a look at the volumetric data, volumetric-based registration projected on the surface. So this is an example of aligning central surface together on the volumetric data of only 11 subjects. And now we can see that it's not doing in a very good way, because the central surface mass is spreading out everywhere around this region. Now a real case problem could be in this particular area, there is a premotor area 6V, and here is primary somatosensory area 1. Here is other like 3 areas in the middle, and they are actually very close to each other in terms of Euclidean distance. But when you do the registration driven by volume space here, clearly you are very likely to compare them like in the same space, and you're comparing the motor function and somatosensory function at the same time, and it's clearly not what we want. But the surface registration doesn't do it in this way. It's trying to move the surface, and the only deformation is happened within this 2D sheet. You can only move in this direction, so the misalignment is less likely to occur, because if you want to mix up these two regions, you find that there are actually area 4, area 3A, area 3B in the middle is much longer. The surface distance is much longer than the Euclidean distance, so you're less likely to mix them together. And this is the advantage of the surface-based spatial correspondence. And now I would like to just briefly show how the surface registration works. I would use the MSM as an example because it's the key surface registration adopted in the HCP pipeline. So the surface feature could be the folding patterns or the functional patterns, and they can all be used to drive the surface registration. And here I'm using a folding-based registration as an example. This is the curvature view of a moving image, and now our goal is to fit align this moving image to this target image. The first thing we do is to impose them into icosphere, because in this icosphere, it simplifies the calculation of the geodesic distance. And on this icosphere, there's a number of the control points in this red grid. It's a low resolution control point. We're just trying to move this control point to the surrounding binary resolution target locations. Once we choose where to move, this grid will move here. And at the same time, the metrics on the graph also move like this. It changed the shape. And now we're just trying to maximize the similarity between the moving image and the target image to make them as similar as possible. At the same time, we also want to impose the regularization on the deformation field because we don't want it. We want the control points, neighboring control points grades, they all move in a similar direction. If they move in an opposite direction, it will end up like this. It's going to reach beyond the limit of neurobiology. So here clearly there are two neighboring control points trying to go in different directions. And now it's trying to squeeze this gyrus into nothing so that it will disappear and match better to the target template. Actually, we can't let it happen because you can't just change the structure of your moving image, right? This is how a surface-based, folding-based registration should work. So after the movement, all of the fold circle pattern should move sharply at this template marked with these white dots. No extra distortions that reach the unrealistic limits. MSM not only allows us to drive the registration using the self-fold depth, but also you can put into multimodal features in it. Just now here giving you an example of the MSMO. It's used in the Nature paper of the HCP parcellation. So it's trying to get all of these features into the same space so that we can use this multiple comprehensive information to parcellate the brain into different cortical regions. So here it first drives the registration using the self-fold depth. Then it's trying to drive the registration using the myelin structure marked by T1 over T2. And then drive it with the resting state network to gain more functional alignment. And after doing this registration, if you calculate the group average functional activities, like this is the language story task, you will find that MSMO works much better than the folding-based registration, like a purely folding-based registration, because MSMO here clearly has a sharper boundary in the functional area overlap, and also it gains more higher stats values. And this is the reason of this is because there is a spatial shift between the folding and function in the higher order cortical regions. So although you have already used the surface-based registration to sharply align all the folds together, they are roughly in the spatial correspondence. And then here in the primary functional regions, like the primary visual cortex or the primary motor, they are already the function areas aligned very well. But in the higher order function regions, like the very complex functions, like this Broca's area here is responsible for language processing. It's quite complicated. It's usually a shift between the folding and function. So the cell code landmark doesn't mark sharply at the boundaries of these kind of higher order function regions. And now, if we want to compare functions, or we want to do group average of these functions, a purely folding-based alignment wouldn't solve the purpose. What makes more sense is to gain more functional alignment together and gain you more specific power. And MSM make this possible on the surface. Now, you know that the whole thing is manipulated on the surface. So to be able to benefit from this, we should first enter through a surface pipeline. Now I'm using the preserver pipeline as an example. The first that you choose, a structural modality that can clearly shows you the tissue contrast around the cortex, and you try to fit tessellated mesh on these inner cortex first, marked by the white matter and gray matter boundary. After you get this, you smooth it and you inflate it to the outer surface of the gray matter. Then you get this inflation view where you can look deep inside into the cell seed, which is very important and usually ignored in the anatomical point of view. Then you project it onto a sphere to simplify the calculation of the geodesic distance, and this enables you to learn the spatial correspondence between all the subjects, so you can perform the population level comparisons later. And during this pipeline, FreeSurfer also outputs you a number of features like the cortical thickness here, the curvatures or the circle depth to show the folding patterns. And these can only be accurately aligned using this kind of surface pipeline. So all of this done is using this single command, FreeSurfer reckon all, where you just need to give it a T1 volume. It's highly recommended to use the 1 millimeter, at least the 1 millimeter isotropic T1 volume data to get a good quality of the surface. And you can also, it's also recommended to use the T2 volume as well for a better like complementary to the T1. Then you can get a better hill surface here. After we have all the surface, now we want to enable the easy visualization and calculation on surface. This is what we want. So all of this done is using a software called Workbench. I'll show you later how to use it. But this software can provide you with a view that you can visualize your surface and QC results and just have checks on these data. But also it gives you a number of the useful commands for you to do some basic operations on the surface. It's very useful. To be able to do this, first we need to change the format from the FreeSurfer output style because it only allows you to do things in FreeSurfer. You change this format into the HTTP output style using this simple command, simplify reckon all. You just need to give it the output directory of the FreeSurfer, and then you will output all of these data for you, that you can build them into the Workbench. So here there are 3 types of file we're going to use. The limpid file, I think that most of you already know what it is. And the gifted file is for the surface left hemisphere or right hemisphere. So for the gifted file, it has several types. It has a surface gifted, contains the mesh file. Basically, it's the XYZ coordinate of the, that it's a mesh, so it has a lot of point clouds. And also it contains the edges between these points so that you form a mesh here. And the metric file is saving the scalar files, like basically any kinds of cortical features or like time series can be saved in this metric file. And the label files are integer data, like parcellations, the region of interest can be saved in this file. SIFT is a more compacted version of the brain. So you can put in the deep gray matter volume data on it. And also it has both of the hemisphere on surface. It also have a label file, a scalar file, time series file, and the dense connect file, which is the connectivity between any pairs of locations in the brain. So you can check, it's not all the city file I'm covering here. You can check if there are other files in this link. So now I'm going to show you the software and the gifted file I've talked about before. This is the surface gifted file. This is the left hemisphere, and this is the right hemisphere structure. From the left hemisphere, we can see here I'm pointing at the white matter surface. We call it white surface. It's basically the boundary between the white matter and the gray matter. And the things above is the hill surface, is the outer surface of the gray matter. So you see that it's clearly inflated from the white. And here in the left hemisphere, you can see this is the mid-thickness surface. It's a kind of surface in between of the white surface and the hill surface. And here is an inflated surface. If you impose a metric file on it, this is the circle depth metric file, you will see that this kind of inflated surface helps you to view deep inside of the cell sheet. And this is very hard to get from the anatomical point of view and always ignored in the previous study. Okay, all this is the GIFT, and this is the safety. I'm showing now a safety file saving the time series. You can go through all the time points from here so that you will see this is a dynamic connectivity pattern. And here you can see in this volumetric view, you can see here is a lot of volume, deep volume voxels. And here is surfaces from both of the hemisphere. And the surface data is better viewed in this surface view, right? And this first, okay, it's all right.
Does it always take an average of all the voxels that go in each of the vertices, or how do you call the surface data points? If you look at functional data,
you mean the time point? If it's time point, then I don't think they are like averaging. Like,
so what is the data that I'm seeing at the surface?
It's the time series data with 1,000 time points. Like,
you're asking how it's projected.
Yeah, so what if there are two voxels, for example, in the range?
I'll show it later with the command. But basically, it's a weighted sampling measurement. So you can resample it inside the cortical region. But I'll show it later in another command.
It's a quick question, like, what's the advantage of safetifying and going to Workbench over just FreeSet, like using FreeView and GH?
Yeah, thank you. I think that I'm not in a good place to say talk about this because I haven't used the FreeView before. But I think the safety file have these advantages, which I'm about to show. So the safety file here is saving you more space. If you're using the safety file, this is the number of vertices and the number of plus the number of voxels here is only the sub-gray matter volume, volume voxels. So we're skipping all of the white matter voxels here. And if you're adding them up, they are altogether called the gray ordinates. And this is all the number of the gray ordinates. And if you compare it with the traditional like 2 millimeter analyze space volume, it's much less and it saves you a long space. Especially in terms of the dense connectum. Dense connectum is any pairs of two locations in the brain, the connectivities. And the resting state time series, it has, if you have like 1,200 time series plus this many voxels, it's much better, it's much more than these safety file. So that's why safety or surface is another like advantage, especially when you're trying to deal with the large scale data like the UK Biobank, like 20,000 data can't deal with this. Yeah, this is why we use the 65. Yeah, for the preserver, probably I think because I don't usually use the FreeView, yeah, so I can't say what is the defects or the advantage of the preserver in a good place. Okay, here is the interesting command, the volume to surface mapping, which is how we project everything from the volume space to the surface. Usually we use it to project the time series data, the function MRI, time series data, or the myelin maps. We get it from the volume, it's T1 over T2, showing like the myelins. Then we project them onto this surface. Usually we need to only give it the volume and the surface you want to project it on. Then here you can specify a number of the options, like how you want it to interpolate or how you want it to resample. Usually we use these reason constraints. We only want to sample the things within this cortical ribbon marked by the inner and outer surface of the brain. We project everything in the middle surface called mid-thickness here. So here is the demonstration of this. this can project any kinds of data. So because I want to like let you know this purpose, I'm choosing a patch data here. It's roughly registered with this structured data of these subjects. We're getting the surface cortical ribbon from the structured data, the T1 data. We have the inner surface and outer surface. Now we got the cortical ribbon, and then we overlay it on these patch data. And now in each of these locations, we form a little like column. Then we try to use the weight. So the closest have higher weight, the further one have the lower weight. And we're just use the weight combined to sample them inside this little column, inside the vertex in the middle point. You understand? That's how we project them onto this surface. And this is the output of the PAT data. You can see that there are high activities in the frontal lobe, high activity in the parietal lobe, and low activity in the usual. And if you compare the left hemisphere from the right hemisphere, you'll find that in the right hemisphere, there's local inactive points. And if we check what is going on in it, you'll find that actually here is a subtle lesion of focal cortical dysplasia. It's very difficult for you to visualize it or to find it in the volume space, but in the surface is quite easy because you only have one view. And at the same time, it's also worth noting that you can't hide any of your results on the surface, and you can't just choose like the best slides you want to show in your result because you see everything here on the surface, so you can't like fool everyone. Another command here I'm going to show is the calculation on surface metric math. You basically just to specify an expression to evaluate like these. You can calculate the average, you can binarize it, you can use the exponential, you can use sine or cosine, calculate these. Basically, just if you can write down the expression, then you can get the metrics. Here I'm using this example of an individual language story task function, right? And I want to get the positive activation of it. So I first name it X, and then I only want the X greater than 0. And now if we have a look, it's here. This is what I gave it. And after we do the evaluation, here is what it looks like. It only gets the red parts and the yellow parts. So it's basically the positive activations. Yeah, there are lots of commands that is very intriguing and very useful. This is the last one I'm going to show. It's calculating the metric gradients. It's very useful if you want to delineate the sharp signal changes. Say, here I'm showing that we have these group average activation map of the language story task. And in this task, the function area 55B is usually activated. And if I want to delineate this region, I can use I can just call the metric gradient, and it will output this gradient map for you. You can see the highlighted things here marked perfectly with the boundaries of this functional area. You can visualize them very easily using this command. So all of these things I've talked about, all of these cortical surface analysis, that together enables the very precise research applications. Again, in this paper, after we use the MSMO, now you can help to identify specific functional area 55B, like here it has low myelination compared to the neighboring vertices. It's activated in the language processing task, language story task. It has very unique connectivity patterns. It's activated by itself, and if you activate the frontal eye field, then it's left behind. You can also view them very clearly if you are marked using the metric gradient, and this helps you to identify these kind of parcellations. And there are also a lot of other areas like defined in this way using the comprehensive knowledge. Okay, is the last slide then. So from all of these things I've been talking about, I hope that you roughly get how the surface-based analysis work and how would it benefit your functional study. Here's just a brief summary of today, everything I've been talking about. And these are all of the resources if you are very interested. I know today's tutorial is just a scratch on the surface. And we are also running a education course in the upcoming OHPM. You can join, or you can, all the material will be made available online then. Now, okay, feel free to ask questions.